Welcome to the lecture series on food microbiology. Today's topic is detection of microbes in food samples. This is in continuation with a couple of lectures which we have been already dealt with, and this will be continued because we have to discuss the various methods which can be used for isolation and identification of spoilage organisms as well as pathogens from food samples. Membrane filter technique. This technique is used for bacteriological examination of water. Remember, water is a main ingredient in most of the food processing techniques. We use water for washing, drinking water, of course, potability of drinking water is the second question. Then the quality of the water which is going to be used for the various food processing techniques also becomes important. So, normally we have tests which can detect the potability of water. Among one of those tests is the membrane filter technique. This technique was developed in Germany during World War II, and at present this is considered as advantageous over the coliform test technique which is normally used. Normally we detect for the presence of coliforms and then confirm for fecal coliforms in water. But this membrane filter technique is supposed to have more advantage than the coliform technique because of the various factors. In this technique, we use a thin membrane filter. This filter disc is made up of cellulose derivatives and it can retain on its surface all the bacteria from the water sample. So, when you have the membrane filter and you have the disc containing the cellulose derivative, when you pour water over it, the bacteria get stuck there, the water will flow through. So, your water sample is passed through the filter disc. The microbe will get trapped on the filter disc, and this filter disc is kept in a petri dish containing the absorbent part, and this is incubated at 37 degrees for 18 to 24 hours. So, the medium here will diffuse through the pores of the filter disc and provides nutrient to the bacteria. After the incubation is over, one can see the colonies developing on the filter disc. The characteristic colonies of the different bacteria would now be studied to determine water potability. So, you now have the bacterial colonies which are growing on the filter disc. Based on the colony morphology, you can identify the organism. The advantages of this technique is a large volume of water can be analyzed in a short time without much expense. The membrane filter can be transferred from one medium to another to differentiate. So, you can now suppose the filter disc contains a huge number of microbiota, a heterogeneous mass, you can now transfer this filter disc into another absorbent pad containing selective or differential media. So, what grows up there would be only those microbes which can utilize and grow based on the selective medium which is present there. Quantitative estimation of certain bacterial types, for example coliforms, can be done using appropriate selective media even when the bacterial types in question are present in small numbers. So, even if the organism which you are looking for is very less in number, even that small number can be isolated and identified using the membrane filter technique. This technique requires much less equipment and therefore can be operated direct in the field. So, suppose you are going for a trekking or you are going to find for an excavation or something like that, and there you want to know the safety of the water present. This would be because we do not require much equipment or any other conditions, this can be easily utilized for detecting the presence of microbes. Disadvantages: high turbidity waters limit volume sampled, high population of background microbiota will result in overgrowth, metals and phenolic components can absorb to filters and inhibit growth. So, if the water is very turbid, then it will be compromised. If the resident normal microflora are very huge, then you will see a large amount of microbes growing up. So, identification of specific organisms becomes very tough, and if the water contains metals or there are some trace phenolic compounds which are present, they will also get stuck onto the filter and that will inhibit the growth. Now, we are going to see some biochemical tests which are commonly used in the lab for identification of specific microorganisms based on the biochemical pathway or the metabolic products formed. A typical example is sugar fermentation. So, in microbiological terms, any component which is fermentable is called as a sugar. Any fermentable substance is called as a sugar, and usually sugar media consists of 1% of the sugar in peptone water along with appropriate indicator. A small tube called as Durham's tube is kept inverted in the sugar tube to detect gas production. Acid production is shown by change in the color of the medium to pink or red. The gas produced collects in the Durham's tube. So, you have the 1% sugar solution taken in a broth, then you have a small Durham's tube put inverted down onto the medium, then you are going to inoculate the bacterial sample, incubate it. Now, if the sugar can be fermented and metabolized by the organism, you might see gas bubbles which get connected in the Durham's tube. You will see a change there. Suppose you have added a neutral indicator. This indicator's color change would also tell you that microorganism has grown in the tube concerned. The sugar which can be utilized can be pentose, any monosaccharide like pentoses, arabinose, xylose. It can be hexoses like dextrose or mannose, disaccharides, saccharose, lactose, polysaccharides like starch and inulin, trisaccharides like raffinose. You can have alcohols like glycerol or sorbitol, glucosides like salicin and esquilin, or it can even be non-carbohydrates like inositol. So, based on the requirement of the organism, in the sense that not all microorganisms would be capable of utilizing any component as a C source. For example, arabinose, xylose, dextrose, mannose, etc. can be used by specific most of the organisms, but there are some microorganisms which will say that I would be able to utilize carbon only if it is in the form of a mannose or only in the form of a lactose. So, the other sugars, even if they do contain C, would not be assimilated by these organisms because they can utilize only lactose. So, this is actually a distinguishing feature telling that this organism, suppose I have a sugar, the solution which I contains lactose, and if the organism does not grow up, then you can say that this is a species or this is an organism which cannot utilize lactose. We need to look for some other C source for it. So, this forms the basis of the identification test. Litmus milk test. This is to differentiate microorganisms based on the various metabolic reactions in the litmus milk, including fermentation, reduction, clot formation, digestion and the formation of gas. And litmus milk test is primarily used to differentiate members within the genus Clostridium. So, Clostridium, as you know, you have a famous foodborne pathogen botulinum. Along with that, you have the other species of Clostridium like tetani, which causes gangrene. You have other species which might not be present in food samples, but which are equally dangerous pathogens. So, to distinguish one isolation whether the genus Clostridium is present, then once you confirm the genus Clostridium is there, then among the genus which is the species which is present. So, for that we can use litmus milk test. This litmus milk test can also differentiate Enterobacteriaceae from the other gram negative bacilli based on the ability of the enterics to reduce litmus. And litmus milk is also used to cultivate and maintain cultures of lactic acid bacteria. So, not only identification of the various species of Clostridium, but the same test can also be utilized to differentiate between gram negative bacilli and Enterobacteriaceae, and also to cultivate and maintain cultures of lactic acid bacteria. Milk, as you know, is an extremely rich, nutrient rich medium which will promote the growth of microorganisms. And the litmus, a pH indicator, is incorporated in the medium for the detection of the production of acid or alkali and oxidation reduction activity. So, depending upon the production of the acid or alkali or based on the change which happens because of the oxidation reduction reaction, you are going to see a color change which will indicate the growth of a particular type of microbe. Fermentation of lactose of milk with the production of lactic acid, for example, is demonstrated when the litmus turns from blue to pink as a result of the acid production. And the accumulation of acid acts on casein the abundant protein which is present in the milk, and this results in the formation of a clot due to the precipitate of casein as calcium caseinate. And with some organisms, the curd will shrink and whey gets formed on the surface. So, this picture actually shows you a series of reactions which will tell you what is the sort of organism which is present in the milk concerned. For example, if you see a pink color change in the litmus milk test, it shows that acid is getting produced. If you see a pink and a solid portion, that is, you will see a white in the lower portion also, but that solid portion will not move, then it is an acid clot. If there are small openings or fissures in the clot, you are going to see gas production. If the clot is broken down to small pieces, that is called as a stomy fermentation. If there is a lower white portion, it is reduction of the litmus. Semi-solid, but you are not going to see a pink color. You see a clear grey fluid at the top, it is curd formation. Clarification of the medium, it is digestion of the peptone and peptonization. Blue medium or a blue band at the top, it shows an alkaline reaction. If there is no change, then it means that none of the above reactions. So, based on the interpretation of what you see, you can identify which is the microbe which is growing there. Indole production. The indole production or the indole test is a biochemical test performed on bacterial species to determine the ability of the organism to convert tryptophan to indole. Tryptophan is an essential amino acid, and some organisms have the capability of converting this tryptophan into indole, and this division is performed by a chain of a number of intracellular enzymes, and this system is generally called as the tryptophanase. So, only those organisms which contain the series of enzymes in the tryptophanase system would be able to convert tryptophan into indole, and because of this change we can now identify that yes, the organism contains tryptophanase system or it does not contain the tryptophanase system. A positive result is shown by the presence of a red or a red violet color on the surface of the alcohol layer of the broth. The negative result will appear as yellow, or sometimes you see a variable result showing as an orange color. In normal food pathogens, if there is an indole test positive, as you see here a pink color at the top, E. coli normally shows this reaction, and this reaction is because of the presence of a scatole, which is also known as methyl indole or methylated indole, which is a metabolite in the pathway of the enzymes which catalyze the tryptophanase system. For example, this is another picture which shows two reactions. This layer, the reddish orange layer or a deep red color, cherry color layer shows an indole positive reaction. This creamy color where you do not see much change, this is an indole negative reaction. For example, if you look for the food pathogens which show indole positive, Aeromonas, Bacillus, E. coli, Haemophilus influenzae, Klebsiella, all those are indole positive. The methyl red test. Methyl red test detects the production of sufficient acid during the fermentation of glucose and the maintenance of conditions such that pH of an old culture is sustained below 4.5. So, this would be shown by a change in the color of the methyl red indicator at the end of the period of incubation. So, what we are looking for is methyl red positive or methyl red negative. You will see a distinct yellow color if the organism is capable of doing the reaction. So, positive, E. coli, Yersinia etc. are methyl red positive. The second tube shows methyl red negative reaction, Enterobacter, Klebsiella pneumoniae etc. So, you can distinguish between E. coli, a pathogen, and Enterobacter, another food pathogen, using the methyl positive, because E. coli is MR positive, Enterobacter is MR negative. Originally, the paired MRVP tests were used to distinguish between the various members of Enterobacteriaceae, but now we use them to distinguish other bacteria also, including actinobacteria, and the quality control for MR test is one, Klebsiella pneumoniae, which is MR negative, the broth still remains yellow, E. coli MR positive. So, this is the standard ATCC strain which we use to test. So, if these two cultures, E. coli ATCC strain shows MR positive, Klebsiella 80cc strains MR negative. This is a standard quality control of whether your test reagent is working all right, because always in any lab the efficacy of the reagent would also affect the quality of the test and the results and the interpretation we make from the results. The methyl red test must not be performed unless the medium has been incubated for a minimum of 48 hours. So, the culture has to be in the medium and incubated for a minimum of 48 hours before you add MR and do the test. If you do the test too early, you might get a false positive interpretation which will not give you the correct interpretation. It is important to use a light inoculum. If the inoculum is too heavy, the bacterial growth will be inhibited and result in an invalid test. So, you need just an aliquot, a small aliquot. You cannot inoculate 1 ml of the sample onto the MR test. You just need a small aliquot. And incubation up to 5 days may be necessary for MR test. The organism has to be present in the medium and growing a minimum of 5 days. 48 hours to 5 days would be optimum for getting a positive result on the MR test. Or even if it is a negative test, that time period is important. Voges-Proskauer test. Voges and Proskauer in 1898 observed the production of red color when addition of potassium hydroxide was done to the cultures grown on specific media. And Hardin, another scientist, later revealed that the development of the red color was the result of acetyl methyl carbinol. So, when KOH was added to the culture, you see a red color. So, this was observed by Voges and Proskauer, but they did not analyze why this red color happened. It was Hardin who discovered that the red color was the production of acetyl methyl carbinol. This VP test or the Voges-Proskauer test is used to determine whether an organism can produce acetyl methyl carbinol from glucose. So, when glucose gets fermented, the metabolic byproducts in the pathway can be different. If your organism produces acetyl methyl carbinol, it will be VP positive. If it is not producing acetyl methyl carbinol, but some other byproduct is formed, it will be VP negative. This can be used to distinguish between the bacteria which is present in the medium concerned. If present, acetyl methyl carbinol is converted into diacetyl. Diacetyl has a typical buttery smell in the presence of alpha naphthol and strong alkali and atmospheric oxygen. The alpha naphthol is not part of the original procedure, but it was later on found to add as a color intensifier by Barrett. So, initially when Hardin did the test, alpha naphthol was not a component, but later on it was seen that addition of this alpha naphthol acts as a color intensifier, and the diacetyl and the guanidine containing compounds which are found in the peptones of the broth then condense to form a pinkish red polymer. So, this tube, this is VP positive, you see a pinkish red hue here, the yellowish broth shows VP negative. So, if a pink color develops at the top, for example, streptococci of the viridans group, listeria, Klebsiella, all these are VP positive. There is no pink color development, it is VP negative. Among streptococci, Streptococcus mitis shows a VP negative. So, Streptococcus viridans is positive, Streptococcus mitis is negative. So, among the streptococci genera itself, VP positive and negative test will indicate two different species. So, that specific identification would be possible by VP test. Citrate utilization test. Citrate utilization is commonly employed as a part of the test what we call as the IMVIC, the indole, methyl red, VP and citrate test results that distinguish between the members of Enterobacteriaceae. So, different members of the family Enterobacteriaceae have different biochemical reactions. Some might be common, some might be different or specific. So, the answer to this IMVIC, indole production, the reaction with the methyl red, the Voges-Proskauer test positive or negative, and whether it is capable of utilizing citrate as a C source or not, this would pinpoint and isolate the sample from the sample. This is the pathogen or this is the microbe which has specifically grown. This citrate utilization test, it is used to determine the ability of the bacteria to utilize sodium citrate as the only carbon source. The medium contains no other C source except sodium citrate. So, from this sodium citrate, if the organism is able to take up carbon, then it will grow. If not, it will not be able to grow there. And then inorganic ammonium hydrogen phosphate is the sole fixed nitrogen source. So, here we are making two definers. The only carbon source present is sodium citrate, the only nitrogen source is inorganic ammonium hydrogen phosphate. So, if the organism is capable of utilizing these two, it will grow up. If not, it is not going to grow up. Citrate utilization can be used to distinguish between Klebsiella aerogenes, which is citrus positive, which is Klebsiella is Enterobacterus aerugine, normally occurs in the soil and in the aquatic environment, and it is also present normally. So, and then you have the gut microbiota, the fecal coliform E. coli. So, suppose you want to distinguish between E. coli and Klebsiella, then E. coli is normally citrate negative, Klebsiella is usually citrate positive. So, among these two you can distinguish using the citrate utilization test. And if E. coli grows up, it means that the food sample or whichever sample you are testing, whether it is water, it contains fecal contamination. This picture shows how the color changes and because of the utilization of citrate. On citrate positive, if on this slant you will see the growth and it will change into an intense what we call as the Prussian blue. The alkaline carbonates and the bicarbonates produced as byproducts of the citrate metabolism will raise the pH to above 7.6, and this increase in the pH will cause the bromothymol blue change from the original. This is actually, this green is the original color of the medium, and it is onto this original green color medium that you are inoculating the microorganism. So, if your microbe is capable of utilizing both the citrate and the inorganic ammonium hydrogen phosphate which is present, because of the metabolic reaction and the byproducts which are formed, acidity will increase, this change of the medium to above 7.6, sorry, alkalinity will increase. Based on this, this will change from this deep green into Prussian blue. So, if the organism is not capable, you are not going to see any growth. This color change is also not going to be present. Even after 3 days or 4 days of incubation, you are still going to see the same green color of the uninoculated agar there. So, this will indicate whether the organism is positive or negative. Now, there are a lot of media which are normally used to identify the microbes which are present from food samples. If you see the medium and its use, you have the plate count agar which is done where we look for the aerobic mesophilic count. We have the McConkey broth which finds specifically coliforms. We use the McConkey broth to identify the coliform presence in water. We have the BGLB where we look for the MPN of coliforms in food. Then you have the crystal violet acidic blood agar which we use specifically the fecal streptococci. Then the violet red bile glucose agar where we look for the enumeration of Enterobacteriaceae, Beit-Parker for Staphylococcus aureus, then dichloran specifically for mold. Only the mold sample if present in the food is going to grow up, you are not going to see any bacterial growth there. Likewise, the rose bengal with chloramphenicol, that agar will allow only the growth of mold and yeast, bacteria will not grow up there. So, if your food sample contains fungal spores, mold spores, they will grow up in rose bengal agar, the bacterial samples would be inhibited because of the presence of chloramphenicol. Then cephaloxime tellurite sorbitol medium, this is specific for E. coli 0157. So, the final confirmation test, once you have identified a sample, if we use these plate, then we can identify the bacterial sample which is present. To summarize, in this lecture, we saw the biochemical tests which confirm that from among a heterogeneous pool of microorganisms which have been isolated from a food sample, how you can pinpoint that it is this particular microbe which has grown up, how do you confirm that test. Then finally, we also saw which are the normal media which we use to isolate and confirm the presence of specific food spoilage organisms, yeasts, molds, or even specific food pathogens, which is normally used in any food testing laboratory. Based on this, one, the question first which I was, suppose you are given a sample of beef sandwich or a vegetable sandwich. Your question is, how will you identify the microbes which are present in it? You have to identify the spoilage organism, the normal microbes as well as the pathogenic microbes. Explain the steps you will take for getting this information. So, step by step you have to tell what would be the series of experiments you will do to identify as well as confirm the microbes which are present in the given sample. Thank you.